All right, let's get right into it. Today, in this explainer, we are looking at an absolute revolution in generative biology. For decades, we've treated biological data basically like giant, rigid spreadsheets. But today, we're exploring a completely paradigm-shifting idea. What if we could actually interact with biology as if it were a language? Seriously, hook your imagination onto that for a second. Imagine looking at a cell's transcriptome, the complete set of RNA transcripts being produced, not as static numerical data, but as decipherable text. What if the complex molecular states that decide if a cell is healthy or diseased were literally just waiting to be read, predicted, and even prompted? Okay, let's dive into this with Section 1, the language of cells. So let's look at the engine making all this happen. It's called TranscriptFormer, and it's a generative autoregressive foundation model, which is a bit of a mouthful, but what it essentially means is that it treats every single cell's transcriptome as a cell sentence. It has actually learned the underlying probabilistic grammar of biology, basically the mathematical likelihood of how genes interact. It doesn't just categorize data, it sequentially predicts exactly which gene comes next and how loudly that gene is expressing itself, entirely based on all the genes it just read before it. To make this super clear, think about the AI tools we already use every day. A standard large language model like ChatGPT looks at a sequence of words and predicts the next logical word to build a coherent sentence, right? Well, TranscriptFormer does the exact same thing, but it looks at a sequence of expressed genes to predict the next gene and its expression level. It's building a coherent biological state. It uses causal masking, meaning it only looks at past data to predict the future, along with expression-aware attention. So it's fundamentally generative AI, but custom-built for the language of life.
And that takes us right to Section 2, 1 billion years of data.
Because here's the thing. For a language model to be truly powerful, it needs a massive library. TranscriptFormer's training data spans a staggering 1.53 billion years of evolutionary history. Now, current models are mostly trained just on human data, which honestly completely misses the conserved rules of biology that evolution has been fine-tuning since the dawn of complex life. In this training set, we are looking at a wild range of life. Everything from simple, single-celled yeast organisms all the way to complex vertebrates like chickens. In fact, Transformers Romano was trained on an incredible 12 diverse species. We're talking malaria parasites, sea urchins, fruit flies, mice, and yeah, of course, humans. Which brings us to this astonishing map. This is the phylogenetic tree of the 112 million individual cells used to train the model. Now, historically, comparing a sea urchin cell to a human cell was incredibly tough because their genes just look so different. But TranscriptFormer uses something called ESM2 protein sequence embeddings. Basically, it acts like a universal translator. It maps all these different genes from across the tree of life into a shared, species-agnostic space, finding the universal concepts that link them all together.
All right, moving into Section 3, uncovering hidden biological structures.
Now, what's really interesting here is that because it mapped this universal language so effectively, incredible properties emerged completely unsupervised. The researchers never told the model which cells were at what stage of development. They didn't tell it which species were related to which. Yet, just by reading the raw RNA data, TranscriptFormer perfectly arranged the developmental stages of sperm cell creation into their canonical order. It naturally grouped cells by their evolutionary relationships. It literally learned the structure of biology on its own, just by finding patterns in the text. And this unlocks some absolute mind-blowing zero-shot capabilities. Zero-shot just means the model can perform a task it wasn't explicitly trained to do. Without any task-specific tweaking, TranscriptFormer can accurately detect SARS-CoV-2 infections in human cells. It can spot complex drug-induced transcriptional changes across 95 different compounds in the Tahoe 100M dataset. Plus, it achieves state-of-the-art performance in classifying cell types, actually beating out major existing models like UC and GeneFormer. Just look at this one metric. We're seeing an F1 score greater than 0.92 when transferring immune response annotations across totally different mammalian species. To put that in perspective, the model so deeply understands the underlying concept of cellular inflammation that if it sees an inflamed mouse cell, it can perfectly identify that exact same inflammatory state in a rat, a rabbit, or a pig. The translation is almost flawless. Let's move into Section 4, the ultimate virtual instrument. This right here is the core of our explainer today. We aren't just treating this AI as some really smart biological hard drive. Researchers are transforming it into a queryable virtual instrument. So the crucial point is that this completely shifts our mental model. Because Transformer is generative, we can actively talk to it. We can prompt it to simulate biological responses without ever touching a physical lab bench. Here's exactly how you'd use this virtual instrument in practice. It's a simple first-step process. First, you prompt the model with a specific transcription factor. That's the biological switch turning other genes on or off. Second, the model acts like a simulator, calculating conditional mutual information. It's basically mapping the mathematical links to see which genes are statistically dependent on that factor. And third, it extracts and generates brand new predictions about gene-to-gene interactions, simulating how the cell's network will actually react. You are literally prompting a cell, exactly like you'd prompt an AI chatbot to write a poem. Now, you might be wondering, okay, but is the AI just hallucinating these interactions? And the answer is absolutely not. The hypotheses generated by TranscriptFormer successfully align with the independent string database. That's a massive, trusted catalog of verified, real-world experimental validations. So the model's predictions are highly, highly accurate. Which brings us to our final part, Section 5, Prompting the Future of Biology. Let's step back and see how this all builds into the future of biological research. By taking 1.5 billion years of diverse evolutionary data, translating it through protein embeddings, and feeding it into a generative transformer, well, we've created an unprecedented tool. We can map cells, predict human disease states, and actually run virtual experiments to see how complex gene networks operate across the entire tree of life. It's an interactive, queryable knowledge base of life itself. And that leaves us with this final, really provocative question. As models like Transformer Prompter continue to scale up, and as our ability to prompt these virtual cells gets more and more precise, is prompting an AI going to replace the physical petri dish? I mean, could the next massive medical breakthrough come not from a wet lab, but from a researcher simply typing the right question into the language of cells? It's a fascinating thought to leave you with today. Thanks so much for joining me on this explainer, and keep questioning.